Hello everyone, so welcome back to our review lesson 3. In this particular video, we will be talking about enzymes and of course we'll be focusing more on photosynthesis and cellular respiration. Those are the standards we'll be covering in this particular lesson. The first one, we're going to talk about what are enzymes, and the examples for the enzymes are the salivary amylase, which is in our saliva, and our digestive enzymes, and all the enzymes that perform different functions in our body uh, are pretty much they are proteins which speed up the chemical reaction. So that is what enzymes are. And any chemical that speed up the chemical reaction is called catalyst. So we call enzymes our biological catalyst. And as you see here in this diagram, and so what do you see here? This is the mechanism in which an enzyme act. And you can see here this ghost-looking thing. If this is an enzyme, and so what happens is we have a chemical. Let's say starch that you're eating, potato starch. When you scoop full of potato starch in your mouth, and when you put it in there, immediately saliva is mixed with the starch, and this is the starch which we call as a substrate. So immediately what happens is the starch will go and bind with the amylase enzyme, and you can see that now it forms an enzyme substrate complex. So this molecule pretty much it stays in maybe a very milli fraction of a second, and immediately what this enzyme do is enzyme just break this starch into maltose, two molecules of maltose. That is what here we see it as a product. So this reaction is so quick, and if you chew this a piece of bread in your mouth for significantly maybe like a 10 minutes, you actually start taste sweet because of the product is formed significantly in high amount since you are chewing it for a little bit longer time. So what enzymes do is the enzymes convert the substrate into products. So that is what enzymes do. Now you can see that this enzyme will go back and pick up another starch molecule right here, and then you continue doing this job and keep on making maltose sugar. That's why you taste sweet, even though you're eating a piece of bread which is full of starch. So look at this graph. How does this enzyme work? And this is the graph. What you see here is energy is released and energy is supplied. And there are two reactions. So what we have here is glucose is combining with oxygen, which is nothing but the burning of glucose, which is cellular respiration. What you're getting is carbon dioxide and water. And what I want you to see is this reaction need a push. You can see that the energy need to be applied to run this reaction. This blue line and the red line, these are two different time where the blue one is using with enzyme and the red line is without enzyme. So look what's happening. And when we are not using enzymes to burn sugar, combining with oxygen, we need to apply a lot of energy. You can see that there's a tremendous amount of energy need to be applied to reach this level to get the final products. And what we call is activation energy. And activation energy is the amount of energy required to run this reaction. See, we need a lot of energy without enzymes. Look, when we add enzyme, the amount of activation energy is tremendously decreased, significantly dropped down to this level. So you can see that it's go up a little bit, same thing, a small push, we get the same products what we hear. So this is what how enzyme work. Enzyme reduce the activation energy. Next, we're going to talk about the factors affecting the enzyme activity. Our body temperature is set at 37 degrees Celsius, which is the optimum temperature for all the enzymes to work properly. And at 0 degrees Celsius, at ice cold, you can see that enzymes cannot function at all. And then it is significantly increased. So here it starts the reaction significantly shoot up. Maximum rate you can see around 37 degrees Celsius. If you heat it up again, and the enzymes will denature and the rate of reaction will drop significantly down. In the same way, if you look at the pH, the pH scale, 7 is neutral and less than 7 is acidic and greater than 7 is basic. So if you look at the graph here, There are three enzymes you can see here. One is the pepsin, salivary amylase, and alkaline phosphatase. And the salivary amylase is working really well in this around the neutral pH, which is around 6.8 or 7. And this is, as I mentioned earlier, salivary amylase works inside our mouth, which is a neutral pH inside our mouth. And if you look at the pepsin, which is actually a digestive enzyme in our stomach, that works great at around 2. And stomach acid keeps the stomach pretty much acidic, and you can see that it works really well at this particular temperature. And alkaline phosphatase is another enzyme, works really well in base pH. You can see that around 9, which is significantly base pH. That is the best for this particular enzyme. So from this, what we can conclude is each enzyme has its own um, optimum pH to function well. And you can see this one is works well in acidic environment. This one is in neutral pH, and this one is in base pH. Another factors that controls the rate of reaction is the substrate and enzyme concentration. Imagine you're adding more substrate. You can see the substrate and rate of reaction. More substrate, of course, the reaction is going to increase, but how long? It is going to reach a point of saturation because all the the increase in the concentration doesn't necessarily affect the rate of reaction. It is going to level off because all the enzymes have been used up here. And the same thing here, if you keep on adding enzymes, what happened? Of course, it's going to increase and then it is going to level off as well. So that's all about enzymes. So now I'm going to switch over to the relationship between photosynthesis and cellular respiration. As you all know, photosynthesis is what green plants do. It takes place in the chloroplast. And as you see here, the cellular respiration, which takes place in all living organisms in the mitochondria. So we're going to look at the relationship between these two, and this is referring to the green plants, and this is mitochondria referring to all living organisms, including green plants. So see what happens when the light energy hits the chloroplast. The chloroplast is going to make sugar and it produce the organic molecule, which is glucose and oxygen. Of course, this oxygen is going to release back to the atmosphere, and those oxygen we are going to breathe in. We means all living organisms breathe in. And also we get this organic molecule from our food that we eat. We are going to burn this glucose molecule, which is organic molecule, in the mitochondria and produces the ATP. We release the energy out in the form of heat and also mechanical energy. So you can see that the energy enter into the ecosystem as a light energy come out from the ecosystem or released in the form of heat energy. And see what happens after. What happens during this reaction? Of course, energy is released. Along with the energy, we breathe out carbon dioxide and we pee out, of course, water molecules as well. So this carbon dioxide and water again goes back to the atmosphere and the ecosystem, and then this is absorbed by the plants again. So the carbon dioxide again, plants absorb it and do this photosynthesis and put it in the organic molecule and release the oxygen out. And the oxygen that plants releases out is actually come from the water, and the carbon from the carbon dioxide goes to the organic molecule. So this is how the photosynthesis and cellular respiration is highly related, and without this particular cycle, life is definitely not possible at all, because photosynthesis is what process that produces oxygen for us to live in this planet, also the food for us to live in this planet. And again, all living organisms produce carbon dioxide back to the atmosphere, and water we releases back, and that is absorbed by the plants again, and this cycle continue in this biosphere. And I hope this video is helpful, and we will discuss this in the classroom, and if you have any questions, please let me know. Thank you very much.